Okay. I just want to introduce a little bit about our collaboration, and because initially when I joined the University of Tulsa, every time I give a talk in the presentation, people say, "Are you from OSU?" I say no, because OSU is the main land grant institute, and University of Tulsa is a private institute. It's a small university on the eastern part of the state, and I'm the only guy working on plants. So before that, I work in the Nobel Foundation, and I moved from Australia, so almost 18 years. If you want to know more about my lab, we work a lot of different field crops in Oklahoma. And one time, I think in spring, from university, no, from USDA, Lai King came and he gave a talk, and he said, "You are doing a lot of work. The Oklahoma State University don't need to hire another applied virologist." so we work with the growers, we work with the county extension. I'm mostly focused on virus ecology, epidemiology, virus evolution. That I worked 3 years with Maryland. And then also started some microviruses since 2017 when I got a Fulbright in work in Japan on microviruses. So you will find all of those things, publication and everything. So this is the graduate student, Caleb Parsley. He was sick, he couldn't make it, but he's the one who worked most of this on this project, on Rosetta screening and all those things. So he's very good. He's local from Tulsa. He can drive by himself, and that make it my life much easier. The team that we have, the Oklahoma team. So Francisco is right here in Penn County. I'm in Tulsa County, and by road, I think it takes around 1 hour 8 minutes. So if our plot is in the Mango, OSU Mango Valley Research Station. It took me 15, 20 minutes, but for him, it will take around more than an hour, 1.5 hours. And since I'm a joint professor there, so I have access to their facilities, and I have so many other plot and forecast station and everything, because of Tulsa University, they don't have any field related program. So this is just to, again, explain that where we put in the Mango Research Station, all of these plots, almost more than 2,000, some of them survive. We currently have around 1,800 something. They were established initially, we had a hard time, and then some rain and flood, but they were, slowly getting up, and now you can see, you can hide in them, hide and seek for the kids. They're huge. We did some pruning. It's really nice. So this is the plan that these are around 20 row, but up to halfway here, this is my area, this is Francisco area. We divided 900 and 900. So, but we work together as just, in case we started the PCR, neither he, neither I can handle 1,800. So we just, the student has to do the sampling, but we all go to the field together. So what we did, once we established in April the second planting, then Francisco said, let's do some inoculation, because on the natural inoculum, we don't know where the mite is there, the inoculum is there. So he brought some from his greenhouse, all of these rosette, and we did some clip, we just cut those small, that will take 2 or maybe 24 hours or 48 to stay green, and the mites can transfer the virus. Because the idea is to screen all of these 1,800 plus rose accession and varieties to the rose rosetta natural screening. But we have to have some inoculum. So based on that, when we look at it, this is our random sampling from each row. So all of these blue, around 81 sample that we got randomly based on the symptom or whatever we could see, observe. Only one part was naturally infected. This was a tentative of the other. Then we found a lot of rose cryptic virus, which is a double-stranded RNA virus, and it is a latent infection. So I don't have time to talk about this story, but we'll just focus on the rosette at the moment. So we thought we need to have more RRV inoculum, and multiplying the RRV inoculum in the greenhouse also takes time. So I decided that I'm more familiar with the field, the county extension agent. So Francisco said that he will multiply the greenhouse and I'm gonna keep looking for inoculum. And it was not a fun story because it's not a crop. So you don't go anywhere that you can see all the roses at once and you just pick the things. So we were going through different nursery, department store, contacted them. Sometimes we work on the bank, in front of the bank, or, Taco Bell, anything we see. And they would come, what are you doing? We say, we just want to see, because you have to open, take the picture, take the sample, everything. Sometimes we have to buy the burger just to show that we are here and just, So, because I had a previous, another student, he was working on milkweed and she was sampling on the roadside, and then the petrol highway stopper, what are you doing on, why you are picking this thing? So we don't want to be in trouble. Sometime we went to one nursery, we went in Broken Arrow, and there were 2 infected plants. We knew that this is infected. They put it on the side. So we asked the person, what are you doing? They say, we will throw it away. So we want to get it. When we went to the shop, they say it's $25. I say, these are infected, you're going to throw it away? No, we can't sell it. So we bought 2 plants on $50 and just take it to the plant, to the field, establish it there. Then in Lawton, Oklahoma, we went there and we didn't want to show that we are working on this. So we like a customer, and the gardener, we asked him, do you know Rose Rosetta? Oh, this is bad, and like a disease. And we found one showing symptom. And we thought, okay, do you think this is? And he said, yes, that's the one. He picked the plant and just threw it away and said, do you mind we can have it? Yeah, take it, the whole plant. So sometimes you get free plants, sometimes you have to buy it. So that was the thing. So we got around 20 infected plants and we put it in the field, established it in different row randomly in the summer. And then Francisco in April this year, he got some more multiplication ready. And then this time without clipping, we brought the whole plant. So we have almost more than 40 plants distributed nicely. And we just need the mite. I hope some of them, the mite will be there too. But we can borrow some mite somewhere and put it there. But we believe that this summer, there should be more spread in our field. So this is the county that we visited. We collected a lot of sample. Again, we found rose traffic virus, and then also a lot of RV. So far, some wild roses we collected, and we also got RV in wild roses. So that's something unique we are looking into that. So during our collection, when we bring the plants and put it in the pot, some of them, they were ready in the pot. We thought there was whatever I'm talking is, we already published this because we realized that there is a small window, or not small, a big window, because these plants cost $20, $25. And if you have in the landscape, only one cane is infected, and then you remove the whole plant, because the public has that fear that it's gonna go kill all our roses. So what we did, these are the 2 plants that we looked at. One cane was infected here, this one, and we put label it C1 to C6, all the canes in that one, and that has another cane. So on R79, we wanted to see how quickly the symptom in the virus will spread. So we tested on every cane in different time point with the primers by PCR, and we found, with a positive control, we confirmed that. So we did the observation first. That was the infected one, and this is by PCR confirmation on August 9. Then you can see that how quickly it can spread until November 11. We didn't see anything on this plant, any further spread. So it's almost 3 months. So we thought during this time, if the grower or the gardener can get rid of that one, their cane, for the landscaping or other thing, it will be still good rather than remove everything and just have a barren landscaping or garden. Then so this is the the cane that was infected right here. So in the previous slide, we put it in the wrong way, but actually this is the one that was the cane, and there was no further spread to any. In case of R80, all these 6 canes, it was right at the bottom. So see, one was infected, showing symptom. And again, we observe this one to see how far, so by October 18, we saw the other canes was infected. So again, it took around 78 days for the virus systemically going from one cane to another cane. And I was talking to Jane Mary yesterday that in the annual crop, they have one main stem and leaves, and the virus movement is very fast, and they can reach very quickly to the apical meristem. This is a woody plant. So the complication here and systemic movement might be different from the annual herbaceous plants compared to the woody plant. So this is the one that we found that the symptoms move slowly there. So these are all with different RNA we try because we just want to make sure that how many RNAs are keep moving. And that could be another thing that we need to look at it, that the virus need only how many RNAs for successful symptoms and infection. So these are the RNA tested in every time point and every cane we did. And here, for RNA3, we couldn't find any RNA3 in any cane, including this one. So that will be something to think about why it is going like that.
So quick question right there.
Yeah.
2 things. What cultivar? And then what PCR test did you use?
These are knockout and that's RT-PCR. Yeah. No cure PCR. Yeah. So you can see that the arrangement of these canes, the combination. We also check the roots as well in the R80, and they were not infected at all. So then we look at R99. This is another plant, and up to 193 days, we got the second cane, the symptoms on this one. So this was the initial one, and this is the second one. So on R99, if you look at it, normally you have more thorniness in the Rosa rosetta. In this case, this was very mild, no thorniness, and that was also unique best when you compare it with other samples that we got it. So that also was very interesting. And from there, we thought, since we have all of these different collections and different, why not we look at different, either there are different isolates or because the study that have been completed previously, I'm going to talk in a minute. These are all the different samples that we collected, some of them in the past. This year I was in Australia for a conference, and one of the county extension, the nursery guy called me, and I say I'm out of the country. He said I have a lot of Rosa rosetta, and I called Caleb, and that's the one he gave it to us free of charge. So that's a good inoculum. We again transported some to the field and then collected some sample as well. This really, that was gold mine to have that many Rosa rosetta inoculum. So here is the genetic diversity. We wanted to focus that how if there is any genetic variation, because these are the few study that has been done. The last one was by Jane Mary, and based on that, the genetic diversity was very low because all of those isolates either came in the same year, from the same collection, or from the same state. So we hypothesize that if there is more variation, we will have a clade in Oklahoma isolate. So far we have sequenced only 7 isolates, and we did find in Oklahoma clade here. These are completely unique. The other 3, this is the isolate Francisco was mentioning, the T sequence, and these are our 3 other isolates that we collected. Our other 4 isolates distributed with others. So there is some interesting, phylogeny, but we are adding now more isolate, at least another 12, 13 to see the distribution either, because it could be the epicenter of Rosetta, that long ago they are, systemic bottlenecks, chromides, horizontal bottlenecks, all those things. And we might end up with a unique isolate. So this is based on the complete genome, on the 7 RNAs, but we did also on each RNA as well, and we consistently get this clade. But due to the time limit, I didn't want to put everything. So we are up to here, is still ongoing. We also try to purify the virus. We got some success. We got some total protein we can detect in our VLP purification preparation by PCR. That RV is there, but we are still, calibrating and optimization our different sucrose gradient and all those things to purify the virus particle later on for antibody production. So there is a lot of undergrad. We also did some microvirus work in the rose field, collecting all different fungi from roses. We got at least 3, 7, 8 microviruses there, and these are all the different undergrad they are involved. So again, due to the time limit, I cannot talk everything, but we have like a lot of data on this as well. So this is the team. Acknowledge the funding from USDA and all the team. And I will just leave it here. Thank you very much.